pentose phosphate pathway, it's going to be NADP to get reduced to NADPH. Right, so NADP is the electron acceptor. Okay. So this generates a 5-carbon compound now. Right? We've gone from 6 to 5. We've split off CO2. This compound is called ribulose 5-phosphate, and then it isomerizes. This is a rearrangement reaction by which we go from this carbonyl at C2 to an aldehyde at C1. Okay. So that's a rearrangement reaction there. And now we have ribose 5-phosphate, which is what the cell will use for nucleic acid synthesis. So we don't think about that anymore now. So assembly of nucleotides is something that you may hit next term, and then you'll know where this came from. Ribose 5-phosphate here, this isomerase in here, not written down, but in here is the ribulose 5-phosphate that was a step before. Right? It's actually here in the middle. And some of this ribose 5-phosphate, or maybe all of it actually, might get used in nucleotide synthesis. Right? But this pathway generates NADPH. Right? And when there's enough NADPH has been made, you can actually feed back and block the pathway here. Okay? So when that happens, we're not going to make any more pentose phosphates anymore. We have enough NADPH, so we turn off this enzyme. And then what happens is we go through glycolysis. Okay? So we have a regulation here. So we haven't really been talking explicitly about regulation too much yet. But here, it's basically, you can look at it as a feedback inhibitor, because this pathway is to generate NADPH. If we've got enough of it, there's so much around it, it's building up, in other words, then it won't have a strong ability to inhibit this enzyme, but it will have some ability to inhibit it as really as a product inhibitor, or maybe as an inhibitor I find somewhere else on the enzyme. I don't know which it is. But either way, it feeds back and inhibits. Okay. So, in addition to that, we have this partitioning here where we've generated some ribose 5-phosphate. You can make ribulose here, and so there's be some ribose. This could get drawn off, right, to make nucleotides. But to the extent that it's not drawn off to make nucleotides, it gets the carbons here get recycled. Okay, so you can see what's going on here. The glucose is oxidized to CO2 to generate the energy-rich cofactor, the NADPH. And that process is only generating so much NADPH. There's a feedback mechanism by which we turn it all off if we have too much NADPH. But then we've still got all these 5-carbon compounds that get taken in as glucose, and we're certainly not going to waste them. Right? So the rest of this stuff is a recycling. Okay? And so this is called the non-oxidative part of the cycle, because there's no change in redox states. It's just taking the carbon skeletons and rearranging them in such a way as to feed them back into the mainstream of the pathways. Right? And the mainstream of the pathways, of course, is this. Right? Here's glycolysis, glucose 6-phosphate, fructose 6-phosphate. Here's the aldolase in glycolysis, right? Generating G3P. Right? So this you're familiar with already. And so if we're here, we merely need to get back. Right? And so in order to get back, you see there's this complicated set of reactions, which is sort of diagrammed here in terms of how many carbons there are in the molecule. Do not worry about remembering the details of exactly how these carbon steps go. Okay? So it's enough to know, it's enough for us to know that this pathway can produce ribose 5-phosphate. To the extent that it does not produce ribose 5-phosphate, this ribulose 5-phosphate, which is actually the precursor in the pathway, can epimerize to xylulose 5-phosphate. And then these sugars, the ribose and the xylulose, undergo a transketolase reaction, then a transaldolase reaction, feeds off some of the carbon, a transketolase reaction, and eventually we've got everything fed back in here. So we're conserving the carbon. The way this is drawn it's drawn in such a way as to feed everything back to glucose 6-phosphate so we can oxidize more. Okay? So if we want a lot of NADPH, if we wanted to have a lot of NADPH, we're not turning off the oxidative part, and we don't need a lot of this stuff, okay, then we would recycle all of this, and this would keep on going. Right? We could get to back to here. Notice that these arrows are going up. Right? Glycolysis, the arrows go down, but we've also talked about how glycolysis is reversible. Glycolysis reversed is called gluconeogenesis. So for now, you just need to remember that these reactions are going backwards. They get to the glycolytic intermediates, and then they run backwards to get to glucose 6-phosphate. Okay? And all of these reactions can run backwards. Yeah, question?
So that would happen if NADPH was, there was such a high concentration that it turned off, and then the ribose or ribose 5-phosphate would go back. Wouldn't that produce more NADPH?
Yeah, so this would happen when it doesn't get turned off. In other words, if you want to have more and more NADPH made, then you want to keep on stimulating this reaction, these oxidative reactions. It gets made in here, right? So NADPH is splitting off this dotted line here. So if you want more of it to come in, of course you could be getting more glucose in and do it that way, right? But you also don't want to waste this carbon. So if you get to here and you want more NADPH, then you would run backwards up this way. If you get to here and you don't need more NADPH, then you could run this way to generate more ATP. If you don't need that either, then you could do something else that we haven't talked about yet, which is go back to glucose and go up to glycogen. Yeah. So there's all these different choices. Yeah.
So it sounds like don't waste our time with knowing the specific enzymes and the names of.
No, you need to know the enzymes. I'll talk about that in a moment.
Okay.
What you don't need to worry about is, see here it's 5 + 5. A ketolase is a 2-carbon transfer enzyme. You need to know that. So 5 + 5, if you're transferring 2 carbons, 5 + 5 is going to go to 7 + 3. An aldolase is a 3-carbon transfer, like the aldolase in glycolysis was 6 to 3 + 3. So 7 + 3 goes to 6 + 4, so this was 3. 7 minus 3 is 4. Right? So this is a 3-carbon, this is another 2-carbon. Okay? So what you need to remember is ribose 5-phosphate is formed, it isomerizes, it polymerizes. These 2 sugars, which we don't need to worry about the structures of either, then undergoes some reactions involving 2 and 3 carbon transfers to recycle into the main intermediate pathways, the main pathways for glycolysis, and going the other way, gluconeogenesis. Yeah?
So when we were talking about glycolysis, you mentioned that fructose 1,6-biphosphate is like where the cell goes all in on glycolysis.
Right.
So can we just ignore that and go backwards? Because I thought that was like a...
Well, it hasn't gone all in here, right? It's only gotten to here. It hasn't gone all into glycolysis. It goes this way. I mean, I guess, yeah. So what I'm saying is here, it's drawn to go back. I'm not sure I quite understand your question, but if you get to here, you could potentially go forward.
Okay.
So it would be a way to, it could be a way to go forward without having gotten to F1.
Yeah.
Without having gone through that. Is that what you meant?
I just kind of, I guess I thought that when you said it was the cell going all in, that it was irreversible from that point.
Like step 3.
It's generally irreversible from that point. It doesn't mean that the reactions can't be run backwards.
Okay.
But if you're under conditions where they're going to be running forwards, then they all run forwards. I just meant that there's not feed-off points for most of those intermediates on the way through. Yeah.
So in conditions where you're going back to G6P, then that 3rd step from like at fructose 1,6-bisphosphate, that then it will run in reverse.
It will run in reverse. Yeah, the irreversibility of the glycolysis pathway after F16BP is formed. I just meant it to say that the intermediates don't feed off into other pathways. They just go all the way through to pyruvate. Whereas before that, fructose 6-phosphate, glucose 6-phosphate, then there's glucose 1-phosphate, there's the mutase to go back. Those things can partition around, and this is one of the ways in which they do.
So it's not a thermodynamic, you're